Okay, good morning, everyone. Welcome to another exciting talk in our seminar series. So today we have Fatih Dinç from Stanford University, who is a PhD candidate at the Department of Applied Physics, working at Schnitzer Group, and he also takes part in Stanford's Mind-Brain Computation and Technology Student Membership Program. He received his BS degrees in physics and also in electrical and electronics engineering from Boğaziçi University in 2018. And he also holds a physics master's degree from the University of Waterloo. He conducts research at the intersection of computational neuroscience and machine learning. And today he will talk about using artificial neural network models to explain the workings of the brain. Thank you, Fatih, and the stage is yours.
Thank you so much. I'm actually very excited today to be here, and thank you so much for allowing me to talk today as well. So here's the thing. I had planned to give a slightly different talk. I actually had prepared a different talk like a few weeks ago. But then as I was going through the talk today, I thought it was boring, or at least like it was boring to me at this stage of my knowledge. So I thought I would just change it a little bit. Thus I might be a little bit rusty in the sense that, it's going to be one of my like second or third rounds of this talk, but I hope it's going to be fun. My main goal today is to give you guys a flavor of how to use machine learning in computational neuroscience. But here, at least it's a different field than the ones you guys are probably used to, in the sense that our interest is in applying machine learning to understand the workings of the brain, rather than like being able to predict something really well, or using the most advanced method to basically do inference. In fact, as you will see throughout the talk, one of the biggest issues we have in the field is the low amount of data. Literally every day we are supposed to fit a curve in a like 1,000-dimensional space using only hundreds of like samples. So it's the opposite regime of what a usual machine learning researcher is used to work in. And I hope it's gonna be fun. Oops, sorry. Yeah, so before we start, I just want to show you guys this video. This is a video from the cortex of a mice that's actually doing a task right now. So it's actually moving as well. Or actually it's head fixed, but it's moving with its arms on a roller. And in this video, each dot is actually a single neuron. So we are able to basically visually get the information of like neural activities from the brain. And this is a spectacularly good movie, by the way. Not all of them are this nice looking. But anyway, and today I will be basing most of the discussion on an open question in the field of neuroscience. It's the question of short-term memory, how it's maintained, where it is maintained, and what are the underlying mechanisms at play in maintaining it. And then I wanted to start, in this case, with a small demonstration of what a short-term memory is, in this case, what a visual short-term memory is. So I'm gonna ask everyone to like close your eyes for a second. You are watching this video, and then now I would like you to close your eyes for a very pure second. And if I tell you like, can you please think of the video that I just showed you? When your eyes are closed, can you still remember at least part of the video that you can see? If you do, just like put a thumbs up. If you don't worry about it. See, I see some thumbs up. See, so what we just did is called recall of the short-term memory. Even though you are no longer seeing the video, you can still recall it. So now close your eyes one more time, and this time think about a pleasant memory that you had as a child or whatever. Any memory that gave you joy. Now think about the video again. Can you think about the video again? Can you see the video? Can you see the neurons? Just a little bit, even if like not all of them. Can you still remember part of the video? Just put a thumbs up if you can. Right, so what we have just shown is the maintenance of the memory, and once again the recall of the memory. So you are maintaining the memory in some part of your brain. We don't know which part, it's an open question. And then this maintenance is not disturbed by distractors. Even if I tell you to maintain something else, you are able to still maintain the information that you think is still relevant, which is, in this case, this video. And one last time, I'm going to ask you to close your eyes again, and then tell me from the video that I just showed you, did I show you both hemispheres of the brain, or was it just one hemisphere? If it's both, put thumbs up. If it's just one of them, just put like something like this peace symbol, the two. See, most of you can remember it's two hemispheres. And then the reason why you can do that is not only do you maintain and recall the short-term memory, you can also compute with the short-term memory. And that's the thing. So there are these different functions that we somehow learned how to do that. We somehow learned how to write in and recall the memory, which we think right now is the same function actually. We learned how to maintain it. We thought it was, in some sense, it was actually an advantage in terms of like evolutionary biology. We thought it was some sort of an advantage, so we learned it. And we also learned that we could do computations with it. So these are the parts of the questions. And here's the thing. I'm not going to stop this and then turn into my iPad. Let's see. Can you see my iPad screen?
Yes.
Yes.
Yeah.
Yes.
Awesome. Yeah. So, and these are the things that we learned over the years. And here's the funny thing in neuroscience. We don't know. So we don't know how the recall works. We don't know how the maintenance works. And we don't know how the manipulation of the information works. And more interestingly, we don't know where that happens either. There are kind of theories, and then there are evidence for each of the theory. So someone might just come up and say, oh, it's the prefrontal cortex. Everything is maintained in the prefrontal cortex. And then someone else might just come up with a different idea and then say, no. We have observed causal link with visual short-term memory in the primary sensory areas or whatever. So there are all these different types of evidence in the literature. And that's actually kind of the problem in neuroscience. You kind of need to know which facts you trust more and which facts you need to think more carefully about. And yeah, so that's just like the general idea. And anyway, so in this debate, the way that we are going to make progress is, first of all, have some sort of a task structure. The mouse is going to do some sort of a memory task. In this case, for example, in the task that I'm looking at, the mouse sees some sort of a cue. It's either one of the LEDs that's on during a trial, and then the mouse sees this cue for 3 seconds. And after 3 seconds, the LEDs are completely off. So one of them, you can think of them as being green and the other one being as red. 3 seconds passes, the LEDs are off, and then the mouse has to wait for 3 seconds. So during those 3 seconds, the mouse has to keep in mind, keep in memory what the cue was. And the response period, based on the cue, so one of them tells the mouse to go. So basically it needs to go in this roller thing it's locked in, or it needs to stop, it just needs to wait. And that's the experiment. And if it does the experiment correctly, it gets a reward, and if it doesn't, it gets a punishment, and that's the whole thing. And while the mouse is doing that, we are also imaging the brain. That's actually one of the brain areas. So this red dotted lines, these are the boundaries of the primary visual cortex. And all these green dots are cell filters. Those are actual individual cells that we identified in these movies. So basically what we have access to in neuroscience, and this is going to be the main idea throughout, is we have access to the behavioral readout, what the mouse is doing at each point of time, and neural readout, what the neurons are doing at each point of time. More specifically, we will have the neural activities as a time series. This is a step on its own. There are machine learning algorithms being developed for extracting neural activities from movies. And we actually put out a recent preprint on arXiv very recently on this, but I'm not going to be talking about that. This is actually a very interesting greedy optimization problem on its own. And if you're interested in that part of the research, that's also very helpful. But today is not about this. But yeah, so the main idea is we have neural data and we have behavioral data, and we want to make sense of this data. We have to test hypotheses using that data. And the earliest work on the short-term memory, on the visual short-term memory, actually dates back to 1970s. It's an unsolved problem for like 50 years. And then sometimes you think we are making progress, but it's actually the other way around. You are actually locking the field in a very global minimum, that sort of very local minimum, that actually makes it harder for the field to reach a global minimum. And this is one of those cases. So in 1970s, people started experimenting with the monkeys, and then they put electrodes in the brain, and then they were observing individual neural activities. Keep in mind, around these times, it was actually a groundbreaking idea to do like logistic regression or linear regression on the data. So at this time, people were just looking at the data, and then they were asking, is this neuron firing more or less? And that's like the most computationally intensive questions people were asking at the time. And then they saw that when the mouse was keeping something in memory, the prefrontal cortex neurons were firing more strongly. And then they were firing persistently. They were just like dun 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 throughout the whole delay period. And that kind of became a hallmark hallmark identity of short-term memory for almost 50 years now. Although these ideas are now being challenged, and we are going to be talking about that. But yeah, so it's 1971, and it's literally just monkeys where people are just looking visually which neuron is active and which neuron is not active, and that's basically it. And today we have much more insight on possible theories that can explain how memory is maintained and recalled and etc. And I will talk about them later in this talk. I just want to give so far like a very broad overview of what short-term memory research entails. But first, we are going to talk about the theory of recurrent neural networks, how this theory developed actually in the sense of neuroscience, how people are using recurrent neural networks in the neuroscience research, or in this case, particularly trying to understand how the brain is connected, yada, yada, yada. So the first work that is actually very influential is from 1988. It's two physicists who discovered that if you tune a random network, if you initialize the random network with a specific synaptic connectivity, you can actually have spontaneous activity even though there is no external input. So let's say you have a set of neurons which are connected to each other in a random way. And the finding that these people found was that as long as the connections are drawn from a Gaussian distribution, let's call this G naught, and then as long as this G naught is larger than some G critical, then you would have spontaneous activity in the network, even though there is no external input to the network.
Sorry, question. Are these artificial networks or real neurons we're talking about?
These are artificial networks. Okay. These are random networks that are connected and then evolving in time through linear, nonlinear, the most vanilla dynamics. Does that make sense? Yeah. So this was from 1988. And then this was actually a very interesting idea because indeed, what people had in mind was the spontaneous activity in the cortex. Even the brain, when the brain is not doing anything, when the mouse is sleeping, we observe spontaneous activity. How can this happen, even if there is no, if there is minimal input? And this is actually one of the things that these people proved, kind of gave us an idea. This also actually started the whole field of liquid state machines and equal state networks like 10 years later. So what this inspired in machine learning was that if you had many neurons that are connected to each other, and then you give input from outside, there will be some chaotic, and I'm going to put a big exclamation mark on the chaotic part, there will be some chaotic trajectories happening inside the network, but they are not stochastic, they are chaotic. What this means is that these inputs will drive the network through some high dimensional space, some high dimensional neural activity. But at any point in time, if you want to output some sort of input specific information, you can do that with decaying accuracy with respect to the initial point that you started. So basically you give an input and you don't train anything inside the network. You just let the network spontaneously be active, and the activity is initially driven by input, but once input is gone, it's still there. It's still echoing throughout the whole network. And if you train a set of linear readouts, you could do that. And you could train actually like a choice algorithm, for example, from these. And you could do this in a parallel manner. And the accuracy would be above chance, and it would decay obviously over time, because the memory that's being kept in this network throughout this chaotic trajectory would decay over time. And that's called the echo state property, if you guys are familiar with those. And here's the interesting thing. I specifically said it's chaotic, not stochastic. And I want to focus on the difference here at this. So stochastic and chaotic. In stochastic case, what would happen is things would be random. You put an information, and the network drives itself through some whatever is whether it's a mechanism. In this case, it's just like the usual dynamics of a vanilla network. And after a while, probably in a very short time, what would happen is the network would go into a random state. So doesn't matter what the initial input is. After like a very short amount of time, the network would completely be random, and you would not be able to tell what the input was. In chaotic, you still have this feeling of randomness where you have two trajectories that are initially very close to each other. And after a while, they are going to be very far away from each other. And in fact, in like short amount of times, in sufficiently short amount of times, the difference between trajectories, the distance between trajectories will be exponentially large. But that doesn't mean that these trajectories are not encoding for something. They could still encode for something. And that's the main idea of the liquid state machines and echo state networks. And this came about to be the reservoir computing. It was like one of the things, like one of the big breakthroughs in machine learning in 2000s. And people actually really liked those because you could basically build a memory model. You could build memory in it. What happened next is around 2013, people showed that you can actually boost that memory capacity using what is called an innate training procedure. In this case, what you are doing is, and this is actually a paper in Nature Neuroscience, it's a neuroscience paper, you are training the network separately to reproduce an innate trajectory of your choosing. You say like, whatever is the initial conditions the network starts, if I give this input, I want the network to be trained such that it will always give me the following trajectory. And then the following trajectory is something you specify. And you train the network, and there's a training procedure which I will talk about in a second, but let's assume that there is a way to do this. You train the network not for an outside target. You don't want the network to keep in mind something specific. You just want it to keep in mind that trajectory that it can reproduce. And what this led to is actually an extremely increased amount of memory capacity. So what you could do is then you could basically train any linear readout, not just one function, but many different functions in parallel from this innate trajectory. And that functions have much more larger memories than the previous random networks, the reservoir networks, the echo state networks. And more interestingly, this fair network that they are comparing to is actually a network which is trained with respect to a target. So even though you had a target, instead of training to a target, if you just train something innate and then train a linear readout for that target, you get much better memory capacity, which is insane, right? So you want to do good at the job, and you train the network to do well on the job through back propagation through time or however you want to do it, and then you get worse memory than if you were to just train network to be stable and then train for the job. So that's like the main point that this paper has made in 2013. And that was actually very important for neuroscience, because we always thought as like networks doing a specific job. In this case, people started thinking about networks as like reservoirs where you have these trajectories which are stable and robust. And many different jobs are being done based on these trajectories. They are using those trajectories to do many jobs in parallel. So, and that actually answers one of the important questions in neuroscience, like how do you test causality, right? If this is what's happening, and if I just shut down a process that seems to be correlated with these like innate trajectories, but I don't shut down this innate trajectory, I would not see any effect. And it actually happens because in brain, you can see like 7-fold, 8-fold redundancy of information. regarding a task. Like it starts, like for example, a visual task. You want the mouse to keep in memory some sort of like a visual cue. It comes to the primary visual cortex, but then it goes into the whole, like the whole cortex that I just showed you, like the whole brain. And you can see actually the information spreading from the visual cortex to all the other areas. And the memory, like the mouse keeps that information in all those areas for a very, very long time. But then the question becomes like, which area is actually keeping the information that's causing a link to the task accuracy? And which area is just keeping the information because it just happened to basically get this trajectory started that gets triggered by this set of cues, right? So it could just be a built-in network, built-in function. Whenever something, the brain sees something, it just starts like these different trajectories that are starting everything in parallel, but you just need one of them. You don't need the rest, but the rest is still there and you can still see the redundancy. So that's actually one way of thinking that started with these types of research. And people are actually looking for being able to like distinguish between those types of questions. But yeah, so moving forward, one other thing you can do. So beforehand, we were training to match the neural activities that we want network to have. One other thing you can do is you can not worry about neural activities. You say, I don't care about neural activities. What I want is this network to solve this task. So you can literally take the network, treat it as a monkey, and say the network, okay, learn to do this task that we made monkeys do in 1970s, in 1980s. And then you look at the neural network, you look at the properties of the neural network, the distributional properties, like how the synapses are connected, how well is the memory encoded, yada, yada, yada. And you try to understand how the brain might be solving those types of questions. And what you can do is you can regularize the network. You can regularize many different things in the network. You can say network, you can basically put constraints. You can say, network, you cannot have more than this amount of activity. Network, you cannot have more than this amount of synaptic weights. Network, I ask you to not have as much activity, and that's actually a very important constraint because some parts of the brain they fire crazy, like they have crazy high firing rates, but cortical neurons they don't fire as much, they fire sparsely. So one idea is to put sparse penalty on the neural activities, and then another thing is there are parts of the networks that are not as much connected, so you could also put sparse penalty to the connections as well. So you can try these things and try to understand which leads to a better task performance. And then that kind of tells you about like how these networks are solving, and then you hope to be able to basically generalize to how the mice are solving the task or how the monkeys are solving the task. And I'm going to talk about why these types of research is important to be able to generalize beyond just neural networks in just a second. And why we do computational research if you could just do the experiment, right? What I'm trying to say is we cannot just do the experiment in many cases. And then another set of rules that emerged over time, and this is 2019, again, Nature Neuroscience paper, is local learning rules. So remember the reservoir models, the liquid state machines or the echo state networks, they were not good at keeping memory. And then it turned out that these innate training algorithms, they're also not that good. Like they are better, but they're not that good. And even better types of like keeping memory came about by basically applying these local learning rules, in which cases you are actually learning the synaptic efficacies. So you are not just learning weights in this case, the synaptic efficacies also change over time. What does synaptic efficacy mean, right? Let's start with that question. So usually, if you have one neuron, let's say neuron I, projecting to neuron J, let's say it's sending a synapse to neuron J, you would model the activity, the effect of the, so basically the way that the neuron I affects neuron J as the following. You would say the firing rate of neuron I is multiplied by some weight matrix, which is WJI, basically the weights from I to J. And then you would take this through a nonlinear function, which is a thresholding function for us, right? Because neurons fire after some sort of current they receive, and they don't fire if they don't receive enough current. So that's one way of looking at it, but there is more theory behind that as well. And that's basically it. So this is the input current. The input current to neuron neuron J from I would be omega Ji and Ri, and you would basically sum over all the I's, all the neurons that are giving current to this neuron J, so basically all the other neurons, right? What this network does is it tells you, okay, I'm gonna divide these weights into two parts. One of them are actual weights, actual synaptic weights, and the other one is what I'm gonna call a synaptic efficacy, which is once again Ji. But the synaptic efficacy changes over time. If I use neurotransmitter, let's say this neuron fired, this neuron I fired, now it has less neurotransmitters. So it's either less likely to fire again, or it's more likely to fire again, depending on whether it was a depressing synapse or facilitating synapse. Because what happens when neurons fire is they lose neurotransmitters, that's correct, but they can also gain a utilization rate. They can also be more easily utilized next time. And that actually leads to a synaptic efficacy, which is multiplication between the neurotransmitter utilization, basically the ability of the synapse to utilize neurotransmitter next time, which increases as the neurons fire, but also available number of neurotransmitters, which the network actually has right now, which the synapse currently has. So you multiply them together, and for some of the synapses, that actually decreases after they fire, and for other synapses, that actually increases after they fire. And depending on the type of the synapse, a neuron that has fired once is more likely to fire again or less likely to fire again. So you basically build in this mechanism into the network by some local learning rules. And what happens is, and that's the crazy thing, the network is like 10K more memory capacity with the usual like real life values for these neurotransmitter utilization, decay rates, or whatever, right? So the network that was able to keep something in memory for like 1 second can now do that for 10 seconds or so. So these are also very interesting, and these are actually very helpful because we can use these algorithms to actually test whether biologically whether the predictions are correct or not. And I'm going to talk about it just in a second. And actually, now is a good time to talk about it. So I keep telling you guys that we can test these biologically, and you could just say, like, you could just tell me that, oops, if we can test these biologically, why are we doing this machine learning stuff? Why are we training these networks? Here is how we can test them biologically, right? So we image part of the brain, right? We look at the neural activities, and then we identify that, let's say this neuron, let me just put this neuron, and this neuron are very important. Let's say that we have 3 different theories, and depending on what happens when we silence or activate these neurons, we can differentiate between those 3 different theories. In the past, like until last year, what people could do is they could shut down the whole areas. They would shut down visual cortex, or they would shut down the ACC, or like primary, what was it, like PFC, prefrontal cortex, or whatever. And that's called optogenetics. You would just shine light, and then there was some gene expression happening in the area where if you shine light on top of it, it would just shut down all the neurons through like some gates being opened or closed or whatnot. There is some biology going into it. I'm not an expert in there. And what we have done in the lab is actually we can target individual neurons with light. So we can actually shut down individual neurons or excite individual neurons. So you have theories, 3 different theories. And those 3 different theories give you 3 different predictions for when you shut down this neuron, and this neuron. So you can do that. You can basically build the whole idea. So you build a whole pipeline where you initially image the brain, right? So you basically image the brain, and then you get the neural activities from the pipeline, from the brain. So get the neural activities, and then run basically any type of hypothesis testing. And this is where recurrent neural networks come in. Pick cells to target that will let you that will basically let you decide between the types of theories that are more plausible and then target them. The bottleneck is here. This is expensive. This is hard. Like, you have to do surgery on the mice. You have to hope that the mouse survives the surgery. and then you have to come to the same field of view when you are imaging the second time, and then you have to express the right amount of genes at the right positions, and then you also have to be able to pinpoint your lasers there. And keep in mind that whenever you are imaging the brain, you are also damaging the tissue. So you have to do all of these things in a very short amount of time, because you are literally shining light onto the brain when you are imaging it, which kind of damages the tissue as well. So there is these constraints, and it's actually very expensive. Like a single mouse is much more expensive than paying my tuition, for example. So I am worth less than a mouse, right? So they are very expensive to maintain, and everything is very, so you cannot basically just go around and just like pick neurons and then silence them. You have to come up with a good theory, and you have to come up with a good idea of hypothesis testing before you start doing that. And this is where the recurrent neural networks and all these others algorithms I've been talking about are very helpful for us. And it all has been good so far. Let's talk about the issues here. There are a few issues. First of all, the brain is actually very good at learning. Even if you just like even if you have the same amount of data and train a recurrent neural network, it's not going to learn as well as the brain does, at least as far as we know. The second thing is mice. They are just lazy. You cannot make them do tasks, like they cannot basically perform the same task for more than 1 hour. The most you can get them to perform is 1 hour, and that's usually like hundreds of trials. But you are imaging thousands of neurons. So basically, I am asking you to fit a linear, let's just say you want to fit a linear regression, you want to fit a line in 1,000-dimensional, high dimensional space using only hundreds of samples, which is the same thing as fitting a line through a single point, even worse. Fitting a line through a single point is even easier than what we are going to do, right? And these algorithms are hungry. They need data. So if you want to train a recurrent neural network, you will see it's not easy, right? You need to give it a lot of data, and which is why actually the most widely spread version of recurrent neural network research in neuroscience has been not to fit the brain data, but rather to fit to a target output and try to understand how the network solves that problem. Whereas there is some good development in the former part as well. And now finally, that kind of turns our job into inductive bias engineering. What we are doing is basically exploiting all the inductive biases that we know about the brain. Without it, we have no way of hypothesis testing using neural data. We are using this crazy, like we are basically, I'm going to show you an example and you will see that, but without the types of biases, the learning biases that we are using, we would not be able to come up with any of the algorithms we are coming up today. And then one example of inductive bias is from this paper. It's a Nature paper. Took 3 years to review. Actually, it's very funny, from our lab. It's a very interesting paper. And the main point of the paper is how the brain deals with the noise in its coding properties. So there is obviously noise in the brain. There is different types of noise: correlated noise, uncorrelated noise. Uncorrelated noise you can deal with by averaging, but correlated noise you cannot deal by just averaging over neurons. That kind of builds up. So this paper is about how the brain deals with that. And in this paper, what we observe is when you train, let's say, linear discriminant analysis to discriminate between two types of representation in the brain, like the mouse sees one type of cue, another type of cue, how well the neurons encode this type of information. What you see as the trials increase, and then this is, let's say, the information your decoder is giving you, you see a saturation curve. And the saturation curve happens right before, like, this is the end of the number of trials we can have. This is like, let's say, hundreds or so. So you see the information curve saturating, which is good. And you see this only in the case if you are doing a dimensional reduction, if you are using dimensional reduction as a regularization. And that's very interesting, because dimensional reduction is not the way that we usually regularize, right? We would usually regularize using an L1 or L2 regularization. We would put like an L2 penalty on the logistic regression, or, well, for the linear discriminant you could do shrinkage, or you could run like an L1 penalty, but that's not what works for this type of area. And the reason is The reason is we know that the area that this paper is looking at is actually encoding information in a low dimensional trajectory. We know that from the biological structure of the area. So what we are doing in this case is, instead of using the usual algorithm of running the usual penalties, we are running a partial least squares and picking the first few dimensions based on the cross-validation. And usually it's between 5 to 10, which is what's expected, by the way, from the biology of the area. And you are training a linear discriminant analysis from that 5 dimensions. You have 5 dimensions, 100 trials, you will most likely not overfit. If you had 1,000 dimensions and 100 trials, you will most certainly overfit. So that's the main thing in this paper that actually led the people in this paper to come up with results. And this is one type of an example of an inductive bias that we are exploiting in the brain. Yeah, so it's not just enough to be a machine learning researcher. You also need to know something about the anatomy and how the brain works. And the second thing that people have been very careful about is how the information flows in the brain as well. Like in the types of the tasks that you are looking at, do you really need the recurrent flow of the information? Do you need the information to flow recurrently in the task? Or is it okay if the information was just like a single flow? Is the brain using the information and making the decision right away as it requires the information? So that's the type of information this type of question you can ask. And there is actually the answer to this question has also given us a good reason to not use LSTMs and GRUs. So I've been discussing all these like recurrent neural networks that are built from this vanilla activities, this like just the most basic form of units, which is the linear plus nonlinear units, where you have like linear connections coming in, where you have a neuron where linear weights are coming in, and then you basically learn through a nonlinearity, and then this gives you the firing weights. Instead of doing that, you could say, and the purpose of us, like the purpose we are using these networks is to store memory. And you could be saying, why don't you use LSTMs or GRUs? We know that they are better at storing memories, and then you can actually train them with back-propagation through time, right? So why not just use them? And one of the answers is the linear nonlinear network has actually biological roots as well. You can, this paper, this very philosophical paper, actually Rosa Kao is actually a philosophy professor, not a professor of neuroscience or biology or physics or machine learning. This is actually a philosophy paper, not neuroscience paper, but also neuroscience paper. So this paper actually deals with this idea very vividly, and you can take a look at this paper. But the main gist of the idea is the following: if you use linear plus nonlinear vanilla networks, you have more explanatory power when it comes to using that network to explain what's happening in the brain. And the specific work that they are referencing to is using convolutional neural networks to describe the first pass of the visual information through the visual areas. So the visual areas are literally matched with the layers of the network. And this matching explains up to 50% of explained variance of the neural activities in those brain areas. That's a huge number. And the way that we are able to do that is the linear nonlinear type of neurons that we are using. If we use GRUs or LSTMs, we would not be able to do that. It would not be the same thing. The paper explains why. And the second thing is we are also exporting the fact that the first pass of the information through the visual system is actually non-recurrent. The recurrency starts happening when you start getting feedback. So the information first passes through the whole system, and the system says, oh, look, what you just saw is actually an important, keep that in mind. Or like, what you saw is actually an egg, so there's a computer there or whatever. So the first pass is just seeing, and then the second pass is almost like telling you what is important in what you just saw. So before you go into those recurrent parts, you can learn a lot from just the first pass, which is just a single flow. So you don't need to worry about training a recurrent neural network, which is harder to train and more time to train. You can just train a convolutional neural network. See, so this is another way of approaching these problems. You basically narrow them down and then train the most simple network that's going to be less data hungry than the more complicated one by using both inductive biases. In this case, we know that the first pass is a directed flow, and basically just logic, reasoning. So you're starting probably to see that the main issue we have is data. That's like the whole thing. That's like the whole issue. But yeah, so let me summarize a little bit what I've discussed so far. So in 1988, people realized you can have chaotic activity in networks without any output, without any input, sorry, without any sensory input coming into the brain. You just like close your eyes, there is no input, you just like close your ears, everything, and the network is still going to keep going. And that is due to the recurrent connections. Recurrent connections can do that if you have neurons that are connected to each other with strengths that are larger than some critical amount. And that's a phase transition, right? And then 2000s, people use that idea to train only linear readout of a recurrent neural network, which they deemed the LSM, the liquid state machines, or the echo state machines, echo state networks. And then in 2010s, people started training recurrent neural networks with respect to a target through like back propagation through time. And also, so there was like target-based training, which was done with back propagation through time. But then, you know, you have most of the problems that are associated with this algorithm, which is like you have vanishing gradients, you have like you have to truncate it, which kind of destroys the whole purpose. You want to keep in memory some information as long as you can, and if you have to truncate it, then you cannot really train for that. And then there was also, sorry, this was output-based. Sorry, this is not target, this is output-based. But then also there was target-based training algorithms where targets are actually neural activities. So instead of telling the network to train to do a task good, you are telling the network to reproduce these neural activities that I want this network to reproduce. And this algorithm is actually called force. And later, a better version came out and people called it full force. This is like 2009 and this is 2018. And this algorithm is actually more biologically plausible as well in comparison to the back propagation through time. Back propagation through time requires keeping time information, which is not really plausible when you think of the actual networks in the brain. Whereas force algorithm just uses the error right at this moment, and it doesn't really propagate it through some matrix. So you just need to know your local connections to be able to train with the force algorithm. It's more biologically plausible. And it can be made even more plausible by not letting some of the weights to train. Basically saying, I have some synaptic connections that are plastic and some synaptic connections that are not plastic, so you can make it even more biologically plausible, which led actually people to train networks even better than back propagation through time. And in 2008 and 2019, this was the first paper on the subject, and this is like most impressive one, let's say. People started using these local synaptic update rules, which gave the networks even better memory capacity. And that's where we are right now. So we are trying to use all these training algorithms and all these recurrent neural networks. And once again, keep in mind that the main point why we are training these networks, why we are training these recurrent neural networks on these neurons, is so that we can come up with hypotheses that we can test biologically. So, and it's not possible to test every single hypothesis. It's just too expensive. And recurrent neural networks in this case helps us to dissociate between different types of theories by us constraining them with certain types of constraints, or literally just training them and looking at the connectivities between neurons, or just like running them on a novel input and seeing what happens and try to recover that through experimental procedures.
Can I ask a question?
Sure.
Regarding this, you said that force and full force are using local learning rules too, right? So, but you called the other one as the local synaptic update rules. So you are using Hebbian-like learning, I guess, in both cases. But in the second case, you are basically separating this synaptic efficacy, and you're not really training the remaining part or something.
You are
because you
training.
divided like the synaptic weight into two parts. One of them is the synaptic efficacy, and the other one is basically whatever the synapse size or something. I don't know. I guess it's a long-term changing component, whereas the synaptic efficacy is, I guess, short-term.
Yeah.
The difference between full force and this other local learning.
So these actually come on top of full force or backpropagation through time.
Mm-hmm.
You use full force or backpropagation through time to learn the synaptic weights, and then the local updates update the efficacies.
Mm-hmm.
There are two things to learn here, right? You want to learn the actual synaptic weights, which are like the long-term weights, and then you also want to learn the synaptic efficacies, which is whether the synapse can actually utilize that way or not at the moment.
Mm-hmm.
So you want to learn both of them. The local updates help you learn these, or like basically help you update the synaptic weights such that they will adapt to these synaptic efficacies. And backpropagation through time or full force, you can use in either of them, help you train the whole thing. And by basically taking account that there is a connection between these two through these local update rules.
So is there like is the synaptic efficacy is for short-term memory, whereas the other WIJ term is for long-term memory or something? Is there a division like that, or?
No, and the interesting in this paper is actually they don't force the network to learn in a specific way. They just give the network the ability to change its synaptic efficacies, and the network learns to store the memory in the synaptic efficacies. It's something that the network learns to do. But there is no like, so they are actually very connected, right? The synaptic weights are basically what you would have if the network has not been active for a very long time. Then you would have a synaptic efficacy of 1 or whatever. It's like this the baseline value, right? But if the network is active in a like it was active a very short time ago, then the synaptic efficacies are going to change these weights. But the weights are changing in a very much longer time periods, right? They are not changing as short time periods as the synaptic efficacies are changing. So in that case, they might be, but there's no way that we are enforcing a short-term memory or long-term memory function to these weights, basically. This is something the network learns to do. And one interesting thing in this paper is they actually show that if you just need to maintain the memory, the network does not keep the memory in the neural activities. It just keeps the memory in the synaptic efficacies. But if you have to use that memory, manipulate it, use the information in the memory to make a decision. So if it was just like, I show you random dots in this position, and then 5 seconds later, I show you the same dots again, and then you just say, click. Then you don't have any neural information. Every single information is in synaptic updates or efficacies. But if I tell you, okay, so I'm gonna show you dots which are gonna be oriented up, but then if they're oriented up, you have to basically pick the next figure as something that's 90 degrees oriented. So whatever its initial orientation, you want the second test sample to be 90 degrees oriented from the initial orientation. If it's not, you are gonna say it's not, and if it is, you're gonna say it is. So once you put something like this into the task, where the network needs to compute the information in the synaptic efficacies, then you actually start seeing the neural activities encoding for the information as well. So there is this interesting emerging phenomena that happens in these networks, which you could basically compare to long-term memory, right? Because remembering what the rule was is actually something about the long-term memory. Whereas remembering what the cue was a second ago is short-term memory.
Thank you. Thanks.
Yeah, thank you. Yeah, so these are all very interesting. But then let's go back to the short-term memory debate. Recall that we were doing a task and then we were recording neurons. And one thing that we realized, so this is now coming to what I have been doing throughout my PhD years, is we realized that some of the neurons were particularly active, and this is actually the primary visual cortex. This is a very primary area. And you wouldn't expect that. So primary areas, as I said at the beginning, right, you would expect them to just pass the information along to higher areas. But this area, just like a camera, the area that's used to write in the information, it makes sense that you use the same area to recall the information. So what we see is that there are some neurons in this area which are very active when the information is being recalled. And they are specific to the type of cue that you have presented. So this is a hit cell, which is basically the cell that is active and the mouse is moving forward. And you can see that even though the cell was not active during cue presentation, it's active when the mouse is about to move forward. And here's the thing. This cell is not encoding for the motor action. It's not encoding for the executive plan to move forward. Because even in the trials where the mouse makes a mistake, where the cue was to go, but the mouse chose not to go, this cell is active. So the mouse is recalling the memory, but not basically associating that memory with the go movement. At least if that's the hypothesis. And then there are other cells that do the same thing for the no-go trials. So what we did is I used some algorithms. I don't want to go into detail. It's just like this black box where you put the cells, and then there's a whole pipeline. Let's call this pipeline. You can think of me using that. You can think that I would be using recurrent neural networks to do this. I would use linear models to do this. There are some theory going into this, and then it basically gives you candidates. It says, look, these neurons are important for memory recall. So what we did, we targeted those neurons. We said, okay, we are going to target those neurons in the brain next time. So when the mouse was about to recall the memory, we shut those neurons down. And what happened? The mouse could not remember. The behavioral output went down to chance level right away. Even if you just silence 30 neurons, that's crazy. Like the mouse brain, especially the primary visual area, has like thousands of neurons, 10,000s of 100,000s of neurons. You just silence 30 of them, and the mouse completely forgets the memory. That's an actual causal manipulation that you can do. And that's where my job as a machine learning researcher comes in. I have these thousands neurons to choose from, and then I have 100 trials, and I have to use those 100 trials and 1,000 neurons to give the biologist these cells, target them, and the biologist goes in 2 months of experiment, 3 months of experiments, sometimes years of experiments, and then targets them, and then the mouse completely shuts down. What happens next is what we test next is we want to test what happens if I just excite them out of nowhere. I'm just going to excite them, no trial, nothing. You excite them, and then you see the mouse keeps moving. It's like the mouse is hallucinating. It's seeing something that it's not supposed to see. It's saying, oh, I need to go because I'm going to get a reward if I go. Because we are stimulating the cells that are recalling the memory. So, and these data I'm not showing today because those are preliminary data, and I wasn't sure whether I could show them. But yeah, so this is what we did.
Can I ask a question?
Please.
I know very little about the biological side of things. So what I'm curious about is if you make a distinction between cells that are responsible for storing things, cells that are responsible for recalling things, if those two things are different, and cells or neurons responsible for passing or transferring the information. Is there such a distinction? And if that's the case, how do you make sure that these hit cells or CR cells, they're not the transfer or conductor cells as opposed to like being the storage cells? I don't know if this makes any sense to you, but as an outsider kind of trying to digest this as a computer scientist, this question comes up.
Yeah, that's a perfectly valid question. I just skimmed over those parts because it was details, but that's a perfectly valid question. Any reviewer would ask that. Here is what you do. You silence those cells in different times of the trial. You silence them throughout the delay. You silence them after the recall period, like after the mouse has already recalled and has started the movement. You silence the whole area during the delay. And here's the interesting thing. You silence the whole area during the delay, but the mouse still does perfectly. So silencing the whole area during the delay doesn't change anything. So the information, even if it's maintained in this area, it's not maintained through neural activities. It's either maintained through synapses, like the synaptic efficacies as we just discussed, in a silent manner, which I will discuss now, or they are activated in, they are maintained in a completely different area. And there are some other biological experiments we have done, like silencing the area not through not through optogenetics, which is an optical tool, but through giving the mouse some drugs. Basically, the mouse cannot use any neurons in that area at all. Even if you are not targeting them, it just cannot use any area in that, any neuron in that area. And then you start seeing that the mouse can still do the task if there is no delay. So basically, this area is somewhat responsible for the visual memory, but you don't know for what. And then you start going in silencing area in different times of the trial, and then you start realizing, oh, it cannot be maintenance, because when I silence it during the maintenance, it still did the task perfectly. That's how we know that these cells are most likely responsible for the recall. But then that's a good question, because then you start asking, okay, if these are responsible for the recall, where is this information going to? But where are they sending this information to? And then one very good candidate is the motor areas. We know that primary visual cortex actually sends neurons to motor cortex, which basically decides whether the mouse is going to move or not. So one hypothesis right now that we are testing is whether these cells are communicating with the motor areas, and whether telling the motor areas to go or not go. But yeah, completely valid question. There is a lot of biology and computational analysis behind this that I'm skipping. Happy to discuss at some point. Probably not today, because we are almost running out of time. Yeah. And then there are three kind of theories, but I feel like I am going over time, so I should probably wrap up soon. Let me see. I want to discuss these, so I'm just going to skip over the theory part. But what I was going to show in that part is, as a machine learning researcher doing research in neuroscience, we are also so you also have to make decisions that are going to use these algorithms. But the hard part of the job is not to know how to run the algorithms, rather knowing which decisions to make. There is this puzzle in front of you, and you have these methods that many brilliant scientists have developed, and you have data that has its own limitations, and your job is to take that data with its own limitations, match it with the correct algorithms for hypothesis testing, and then come up with testable hypotheses for experimentalists who can use those ideas and then disambiguate different types of theories from each other. And that's the main point of the slides that I just skimmed through, but that's actually like part of the job as well. But yeah, so where we stand with these RNNs, this is actually from a fellowship application I just sent last week. And the next thing that we are planning to do is doing brain-wide imaging multiple areas in the brain and understanding how they communicate with the visual cortex we just showed is responsible for recall of the short-term memory. And what we are hoping to achieve through this inter-area imaging like models, and then training recurrent neural networks to reproduce the neural activities of those imaging experiments, is to understand how information is being shared between areas. We want to understand when visual cortex sends information to where, and then when it receives that information back, or where the recalled information goes to. Those types of questions. And we have already done some preliminary experiments so far, but most of them are done, we are using basic algorithms like canonical correlation analysis, where you look whether the areas are co-fluctuating together, which is a very common thing to do in like fMRI research, for example, if you are collecting fMRI data. And then that kind of told us like V1 is talking with some areas, and then we, for example, shut down those areas during the task, and we saw that the task accuracy decreased to chance level. So those areas were actually part of the circuitry as well. So the next step is collecting imaging data from those areas, train recurrent neural networks to match the neural activities, and then look at the local connections, which neurons are connected to which. But in this case, how are we going to do this training with low amount of data? And this is where what my proposal is about, is once again using inductive biases. So there is bunch of experimental work showing, and this is a causal work, not correlation types of work. These are correlation works. Causal. These are works that have causal manipulations that show that not just the brain is modular, but even the areas are modular. So we know the brain is modular. The brain has these different types of areas, and they are more connected with each other than within each other than with each other. But even within those areas, some neurons are more connected to each other than the rest. So what are we going to do is, let's say this is an area, we are going to create patches within these areas, and then train recurrent neural networks for each of these areas separately. So let's say this like small area, sub area has like 10 neurons. We are going to find the weights of those 10 neurons by connecting to each other only, and then find the weights within these neurons, and etc. Right? Then use that as an initialization for the whole area training. So it's basically finding a good initialization for your network, and then you will connect these sub parts of the areas with each other through new weights that you need to learn. And once you do that, you are going to do the same thing with the other areas. So you're gonna start small. It's almost like a renormalization procedure in physics, if you guys are familiar with that. But the main idea is start small, start creating patches, connect them together, find how they would be connected together if they were only connected to each other, and then start connecting to other parts. And this way, you are basically priming the recurrent neural network to with like basically good initialization, instead of just initializing everything and telling the network, okay, learn everything from like 100 trials. 100 trials is like 100 times like what? There is around 600 data points per trial. So like 60K data points, you cannot learn the synaptic weights efficiently. There's no way you can do that for like thousands of neurons. But yeah, that's for example one way of doing research in this area of computational neuroscience. And I think that's it for me. Thank you so much.
Thank you, Fatih. Yeah, this was really an interesting talk. We are open to questions from our audience.
Can I ask a question regarding the previous slide? So you got basically measurements from the, for example, mouse, right? And then do you have like this model of neurons you want to train? Do they have one-to-one correspondence with the neurons in the mouse? And when you say like training, you mean you are matching the neural activity coming from the measurements with those one-to-one correspondent neurons, or
yeah.
Okay.
Yeah, so here's the thing. When we are measuring, when we are just like getting these neural activities from the neurons, we have no idea how they are connected with each other. That's what makes recurrent neural networks very powerful, because they don't only reproduce the neural activities, they give us a way of reproducing these neural activities with how the neurons are connected. They give us like, this should be the connectivity metrics for this activity to be reproduced from the neurons that we are observing, basically. And that connectivity matrix is actually important. We know that connectivity matrix will not be stable at a single neuron level from split to split. So you train, like you split data to training and test set, and then you like 100 different splits. And then you ask the question, okay, how stable is the weights found for like neuron I to neuron J? Individually, not much. You don't have enough neural data to constrain that. But distributionally, as in like in terms of a distributional property, what's the distribution of the weights across the whole brain area? It's very robust. And the next thing you can say is, what's the current that flows from one area to another area, which you cannot get from neural recordings. But if you have a recurrent neural network, you can find the current by multiplying the activity of the neurons with the weights you found, right? You can ask those questions, and the currents shared by the areas are also very, very robust from split to split, which basically tells you that this area is very likely to talk to this area at this time of the trial. Like you can have for example, a graph where you have V1 to, let's say, V2 current, and this would be time. And what would happen, for example, is at the beginning, V1 would send current to V2, and then you would go down to like baseline levels. And later on, V2 send the current to V1 basically as a feedback curve. And you can actually see those things thanks to the recurrent neural network that you just trained.
But still, I mean, regarding, for example, as you said, there are like hundreds of thousands of neurons actually in those areas. Potentially your recurrent neural nets are using less than that, right? So they're sort of a logical model rather than one-to-one model in terms of, right? Is it, or?
You try to match neural activities one-to-one. That's what the training algorithm is. But you are correct in the sense that there are neurons, for example, you are not observing, which have connections that you should have known about. And that's actually part of the job. That's like part of the job of convincing someone what you find is robust through that issue. Like it is robust despite that issue. And as I said, like that's one reason why the weights found at the individual neuron levels, they are not robust. They change from split to split, which means they are not really the actual synaptic weights. But what you find between areas and what you find as a distributional value. So it's like in physics, right? You don't know where each gas in the room is exactly, but you know their temperature, so you know the pressure that they are doing to the wall. The same idea here. You don't know at a single level how neuron I is projecting to neuron J, but you know at a distributional level what you find is robust. You can test it with experiments that it is robust with causal experiments. People have done that. And then you can say that since it's robust from like split to split, let's see if we can find something interesting using it on areas that we don't know anything about. And people actually did find some new results from those as well. And then they tested obviously with causal manipulation. So that's the whole gist of it. Whatever you do, you have to test it with causal manipulations. If you find some interesting result, silence that area, see what happens. Right? You have a recurrent neural network you trained. Okay, silence an area. Silence the area on the recurrent neural network. Do you get the same type of information? And in AI terms, that's actually whether your network generalizes out of distribution or not, right? You train it on a... Yes, sir?
Thank you. Thanks a lot.
Thank you.
Fatih, I have a basic question. Again, as an outsider to the field, I try to connect my own research to the topics you mentioned today. So one topic that has been explored in computer vision is understanding the intrinsic properties of images, like, for example, image aesthetics. And within our group, like about approximately 10 years ago, we looked into this problem called image memorability, where the task is to predict how much memorable a given image is. So we have basically some data by experiments, like the subjects try to remember which images they have been shown. So thinking from that perspective, like here, you basically mostly talk about these weights, how they evolve over time. But in terms of these inputs, how do you make sure that the input is like equally memorable so that you can measure the recall? So is this something you need to consider, or maybe it's totally out of reach for your experiments?
So input is actually a big question. How you are going to input into the network, because you don't know which neurons network is inputting into, right? Even before we go into those types of questions, like how you are going to give the input, where are you going to give the input to, right? That's actually a big question as well. You don't know which weights you are going to give to each neuron from each input neuron. And I think one idea that people have been exploring in this sense is just randomizing it, just give random inputs to random neurons. But like basically you have two types of inputs, and then you have random noise on top of it. And then at each split of the network, at each retraining of the network, you change the neurons that receive the inputs, right? So you want to basically argue that it doesn't matter which neurons are receiving the input. The results that I am finding from this algorithm or like from this computational thought experiment is robust to how the input and where the input is given to the network.
Okay.
And then obviously you have to like check like what happens if the noise levels is comparable to the signal level. Like one thing that we are doing, for example, like input 1 is, let's say, like something like this, right? And then input 2 is something like this. So like it's just 2 pulses, whereas input 1 is 1 pulse. And then they obviously project to different types of neurons. They are not projecting to the same neurons. And then you randomize the neurons that they are projecting to, and then you also add random noise here. And one argument you have to make when you are using these types of hypothesis testing models is the result you get is robust to which neurons you are giving the input to, and robust whether you change the noise levels or not. Like even if you increase the noise levels very highly, you should be robust. I think that's like how much we can do. I don't think we can do more than that because we don't know. Like,
so I'm just considering this experiment. Like after a while, maybe you change the environment. You show these mice so that the inputs are much novel in the sense that it hasn't seen something similar before. So maybe those inputs might have more effective way to understand these different neurons and connections. So that's
well, that's a very valid question. It's a very open question as well. Like, how are you going to model the inputs? Is a very open and very valid question that I don't know the answer to.
okay. Thanks.
Thank you.
Okay.
Fatih, I have a. Can I have a? Do we have a time to ask a question? I could, or are we closing?
Yeah, I think we can have a couple of
Okay,
questions.
Fatih, again, I'm an outsider as well as the others. I think I'm trying to still digest all the information that you provided, and it wasn't easy for me. So are you familiar with the memory model developed by Atkinson and Shiffrin in 1770s? It's a well-known model in psychology literature, which suggests that you put the information into the short-term memory first, and then you rehearse it, and then it goes to the long-term memory. Do you know anything about it? I mean, it's a very well-known in psychology. It's something that you may want to look at it. Anyway, if you are not familiar with it, I mean, there are more developments happen afterwards, like people move towards to the working memory afterwards. I guess there were some experiments supporting this Atkinson and Shiffrin model, and there were some experiments which were showing the pitfalls of this model. So I was just going to ask like how this RNN or artificial intelligence really information about this model, how much it, it's still a debatable model, although it was developed in the 70s. It is still one of the best models on memory. So I was just going to ask you how much progress made in AI literature on this regard. But if you're not familiar with it, that's okay. So don't worry about it.
Yeah, so I mean, I'm only familiar at a level of like a very beginner student. I don't think I would be like able to give a satisfactory answer there, mostly because it's a completely different field to associate short-term memory with the long-term memory, like how it becomes long-term. It's a completely different question that's very hard to answer. And like as I said at the beginning, right, we don't even know where the short-term memory is maintained, recalled, and even this like maintenance, recall, and executive function.
Yes, I mean, like for example, in Atkinson, I'm interrupting it. In Atkinson model, that they claim that the short-term storage has a decay model. It has a decay model. In time, like you forget or you lost the information, or it's very quickly replaced. So, but this is still debatable, whether there is such a decaying function exists in the short-term memory or not, is still debatable. And also the later studies show that like the gateway to long-term storage is not just a short-term storage. Like especially neuropsychological patients, experiments on neuropsychological patients show that although they don't have a good short-term memory, they have a good long-term memory, for example. So that means that's not the only gateway. So I was just wondering like how AI could maybe like the models, AI models could respond like based on the neurological data, how it could respond to this kind of question. So, but if you're not familiar with it, I understand. Yes, sorry about it.
Yeah, I mean, yeah. So that's what I was trying to build up to. That's really not my line of research, but there are people in my lab who are doing like long-term memory, engram cell type of research where they could use neural data and then they use these like decoder encoding and decoding ideas. But those types of questions are a little bit harder to like explore with these types of recurrent neural networks and yada yada yada, because you need to know more than just the like connectivity. Like, so here's the thing: the more you need to know about the synapses, the more your manipulation goes into manipulating synapses, the harder it's going to be biologically. If you can explore a question at the neural activity level, that's an easy question to explore because you have easy causal manipulations that you can do. But if you want to manipulate synapses, and which is why it's harder to discuss long-term memory with these types of models, is you will not be able to test those models because we cannot really like silence specific synapses. That doesn't exist. We cannot even in a large scale visually see like which synapses goes to where. If you want to do that, you have to use different types of imaging technologies, and they have their own limitations. So that's a completely different field, completely different assumptions, and completely different issues in terms of like data storage and data manipulation, and in the end, the check of like the hypothesis that you want to do, right? That's why we focused on this problem, which we can answer most of it at the neural level, neural activity level.
Okay, thank you.
Okay, thanks, Fatih. We are a bit over time, so let's thank our speaker again. So it was quite interesting to learn these stuff from you.
Thank you.